And I'm going to talk to you about immortal cells. So, well, first, let's start by defining what a cell is. So, a cell is defined as the basic unit of life that can replicate independently. And in advanced organisms, like in human beings, a cell consists of a nucleus that contains our genes, a cytoplasm that contains our organelles, and all this is surrounded by the cell membrane. So every cell has a cell cycle, which is very tightly regulated. And I'm not going to go through all the different phases of the cell cycle, but basically there's a phase called mitosis, where the cell will divide and will give rise to two identical daughter cells. And this is very well regulated, because we really need to make sure that the two daughter cells are identical to the mother cell and have the same genetic information. But our cells don't only need to divide, they actually also need to die. And that might sound surprising, but it's very important. And every day we have cells in our body that are dying. And we want them to do that, because if they keep just dividing, well, you will end up having some disease, which I'll come back to that. So the process of dying is also very tightly regulated, and it's known as apoptosis, or programmed cell death. So apoptosis comes from the Greek of falling off, as leaves falling from a tree in autumn. So this is a normal process. Trees lose their leaves, and we also need to lose some of our cells. So apoptosis is essential for life during embryonic development as well as in adulthood. As I said, it's very important that we keep this balance between cell division and cell death, because if we don't have this balance, we lose our health. So, for example, if we have too much cell division and not enough cell death, then we'll have some disease like cancer or autoimmune diseases. On the other hand, if we have too much cell death and not enough cell division, then this can lead to neurodegenerative diseases like Parkinson or Alzheimer's. So cell biologists, we have different ways to assess apoptosis in our systems, but one of the characteristics of apoptotic cells is the membrane blebbing, and we can see that by microscopy. So here I put some images of cells that are undergoing apoptosis, and you can see the membrane blebbing. This is a cell under light microscope. In the middle, you can see a cell that has been photographed with a fluorescent microscope. So we can label different structures of the cell with fluorochromes. And here, for example, you can see the membrane blebbing that has been labeled in red. And you can see that as well using electron microscopy. That's one way to assess apoptosis. And there are other characteristics of apoptotic cells, such as DNA fragmentation, that we can also assess, among others. So many people dream about being immortal, but actually a cancer cell is defined as being immortal, and paradoxically this is one of the main causes of cell death worldwide. So I was always fascinated by biology already at school time, and understanding how our human bodies work. And then during my bachelor's degree, I became even more interested in cancer cell biology, and I really was intrigued by understanding how cancer cells become immortal and what can we do to kill cancer cells. So I decided, well, I really want to train as a cancer cell biologist. So I first did my bachelor's degrees in north of Spain. That's where I'm originally from. Then I decided to do a master's degree at the Spanish Research Council in Madrid. Then I moved around, and I went to Switzerland to do my PhD at the University of Basel, because I knew it's a very good university for health science research. And then next I thought, well, I'm going to do some postdoctoral training. So I moved to the US at the University of California, San Diego. And I was lucky to be able to organize my funding so I could choose the lab where I wanted to train in cell biology. And I was also looking for some more sun. So moving from Switzerland back to California was nice. And then at some point I thought, well, it's time for me now to start building my own independent research team, and decided to come back to Europe, apply for a fellowship from Wellcome Trust, and here I am since last year at the University of Liverpool. So I'm going to give you a bit of flavor of the different projects I was involved in during my career. So as a PhD at the University of Basel, I was studying a protein which is expressed in the membrane of cancer cells called CD44V. And it's highly expressed on cancer cells. It's not so expressed in normal cells. And what we found is that cells that express this protein are actually not undergoing apoptosis. The way they do that is CD44 variant will interact with another protein on the membrane of the cell called FAS, which is a death receptor. And by doing that, it blocks signaling of apoptosis. So cancer cells expressing that CD44V protein are not dying. And we were able to make them die by blocking the CD44V protein with a blocking antibody that will release the death receptor and that will lead to apoptosis signaling and kill the cancer cell. And here I'm going to show you a 3D reconstruction of a cancer cell that I stain in green with CD44, if this works. Oops. Yeah. So in green is CD44, in red is the FAS, the death receptor, and you can see the yellow areas. That's where both receptors are interacting, and CD44V is blocking the death receptor. Then next, I moved to University of California, San Diego for my postdoctoral training. And one of the projects I was involved in, it was in understanding a domain of a protein called caspase-8. And well, caspase-8 is known to promote apoptosis through a catalytic domain, but what we found is that the non-catalytic domain of caspase-8 could also trigger apoptosis. So what we did, these are, what you see there, these are cells, cancer cells from neuroblastoma that have been labeled, the nuclei have been labeled in blue so that we can track the cells. And then they have been transfected with that domain of caspase-8 that is labeled in green with a green fluorescent protein. And you're going to see a movie where you're going to see the cells starting to die by apoptosis, if it works. So you see here some blebbing, the blebbing I talked to you about before. So you're going to see there's another cell here dying by apoptosis, another one over here. Here's another one here. So you see all this blebbing, which is characteristic of apoptotic cells. And now I'm currently investigating pancreatic cancer. I chose this cancer because it's really one of the deadliest cancers. And unfortunately, we don't really have good therapy for pancreatic cancer patients. So I decided that I will focus now on this disease and try to understand better the biology of this disease, why pancreatic cancer cells are not responding to the therapy we have, and what could we do to improve therapy for pancreatic cancer. Well, thank you for your attention, and I wish you